there, my name is Saxon Tint, and I'm with Lighteum Medical. We're based in Southern California, and we are a manufacturer of medical components from precious metals and nitinol. And today, I would like to talk about the huge impact that is made by the smallest of materials on medical devices, and that is thin films. So let's get started. So first of all, let's put ourselves in the context of a century of medical evolution. Medical evolution and innovation doesn't happen in isolated breakthroughs. It happens in waves, and each era builds on the one before it. So, for example, in the 1940s, we saw the biggest gains in human health from pharmaceuticals and vaccines. Things like pertussis, which is whooping cough, influenza, and polio vaccines were all introduced in this period, and this fundamentally changed life expectancy across the globe. Then we entered the age of diagnostics and imaging. Technologies like X-ray, ultrasound, CT, they allowed us to see inside of the body without surgery. And diagnosis was no longer based only on symptoms. It became visual and measurable. That set the stage for the next wave, which was implantable electronics. Devices like pacemakers, cochlear implants, and ICDs. They introduced the idea that we could not just observe the body, but we could actively regulate it as well. After this brought minimally invasive interventions, procedures like angioplasty, stenting, shifted care away from open surgery towards catheter-based approaches. So less trauma and faster recovery, which leads to better outcomes. And today, from the 2000s onwards, we're seeing an era of digital and data-driven medicine. So wearables, telemedicine, and AI-assisted diagnostics. It's moving care beyond the hospital and into continuous monitoring, which leads to smarter decision-making. Now, these innovations, they stack atop one another, and they provide the foundation for the next. And personally, I'm very glad that I'm standing on this side of that innovation timeline. So, while we discuss waves of medical innovation, there's a quieter story underneath all of that, and that's the story of biomaterials. Humans have been experimenting with biomaterials for thousands of years. Ancient Egyptians used seashells as dental implants, linen as sutures, coconut shells were used to repair skull defects, and in 1891, elephant ivory was used in what's considered the first recorded hip implant. For most of history, we've relied on natural materials because that's just what we had available. But in the 20th century, a real turning point came when synthetic materials entered medicine. So things like PMMA for dentistry, cellulose acetate in dialysis tubing, Dacron for vascular grafts, and stainless steel and polymers for hip replacements. These materials were borrowed from industry, and they were used because they had the right mechanical properties. And importantly, they didn't immediately poison the patient. This was the first generation of biomaterials, what we now call biopassive. And the goal was simple: do no harm, be as inert as possible. But this approach ran into real problems. Thrombosis, encapsulation, infection, long-term degradation. The perfectly inert material turned out to be a frustrating pursuit. So in the 1970s, something changed. With the rise of molecular biology tools, we could finally see what was happening at the surface, at the level of proteins, platelets, blood cells. So instead of trying to make materials invisible to the body, researchers began asking, what if we can guide this interaction? So that's the second generation bioactive materials. Things like heparin coatings for blood compatibility, drug-eluting stents to prevent restenosis. The materials didn't just sit there, but they actively managed the biological response. And now we're in the third generation, biointeractive and regenerative materials. Materials designed to talk to cells, to encourage tissue integration, proliferation, and even regeneration. This evolution is reflected in how the definition of biocompatibility has also changed, from do no harm to create the most appropriate beneficial response. So this progression has been a key enabler of the medical innovation that we talked about previously. Without advances in biomaterials, especially at the surface, pacemakers, stents, cochlear implants, modern implants, they just wouldn't function the way they do today. So if there's one key idea to take away, it's that the body never sees the bulk of a device. It only sees the surface. And almost every important interaction between a device and the body happens there. Whether it's tribology and wear, blood compatibility and thrombogenicity, bacterial adhesion, drug elution, osseointegration, or electrical sensing and stimulation. All of these are surface-driven phenomena. The core material underneath gives us the structure and the mechanics, but it's the surface that determines how the body responds. So when we talk about biocompatibility, we're often really talking about surface engineering, designing that outermost few nanometers, microns, to control how proteins, cells, bacteria, and tissue interacts with the device. So with that in mind, let's ask ourselves, how can we engineer that surface to give us the response that we want? So one of the ways is with a type of coating known as a thin film. These are thin, solid layers, conformal to your device, and they add functionality to the surface. And the word thin here is doing a lot of work. In modern material science, a thin film might be less than a single atomic layer or several microns thick. The definition isn't very precise, but what really matters is that the layer is precisely controlled. It's sitting on top of a bulk material, and it allows us to change the surface behavior without changing the structure of the device. So, what's amazing is that thin films are really not a modern invention. They've been around for many thousands of years. More than 5,000 years ago, Egyptian craftsmen were producing ultra-thin gold layers for decorative and optical purposes. They would hammer gold films to less than 3,000 angstroms thick, something like 1,000 atoms thick. And these have been found in tombs in pyramids all over Egypt. Skilled artisans today can still hammer gold leaf to about 500 angstroms, which is thin enough to be partially transparent to light. They were essentially practicing thin film deposition thousands of years before the modern processes such as sputtering and evaporation were invented in the 1930s. But since then, thin films are now everywhere. So, for example, in architectural glass, to produce optical control on large sheets of glass, in consumer products to prevent corrosion, and also to visually look attractive, like those faucets you see there. In semiconductors, of course, in packaging, the metal layer on a pack of chips, that's a thin layer of aluminum on a polymer film, and that prevents oxygen ingress from ruining your chips. And of course, in industrial applications too, hard coatings used to increase the lifetime of tools like drill bits and things like that. And of course, in medical devices, things like coatings on stents, implants, things like that. So thin films give us precise control of thickness, composition, and properties. We can customize the outer few nanometers of a device to achieve exactly the interaction we want with the body. Thin films are not just coatings, they're a way to engineer the biointerface itself. So how do we make them? I've split the methods up into two general categories, vacuum deposition methods and non-vacuum. In vacuum methods, you use vacuum chambers, carefully controlled environments, and you create a vapor phase of the coating material. You then put your parts in that chamber and the vapor condenses onto them. Physical vapor deposition makes this vapor by evaporating solid materials. So you got to use an energy source such as heat or a plasma or an electron beam. Chemical vapor deposition makes the vapor using gas phase chemistries. The non-vacuum deposition methods use mostly liquid or aqueous chemistries, combined with either a technique for spreading that chemistry uniformly onto a substrate, or by using another energy source like electricity. The vacuum deposition methods are what we have the most expertise in here at Lighteum, and they have some great advantages. They have very high control of the chemistry, the composition, and the microstructure. And through this control, we can engineer a lot of the properties that we like. So I'm now going to talk through a series of case studies using using thin films in medical devices. Let's talk about one of the most important advances in thin film technology for medical devices, and that's the story of heart disease. So since the 1970s, the age-adjusted death rate from heart disease in the US has fallen by about 2/3, from roughly 760 deaths per 100,000 people to about 260 per 100,000 today. That kind of change doesn't happen because of a single breakthrough. It happened because of a stack of therapies arriving over time. We had better awareness and prevention, pharmaceuticals like thrombolytics that chemically dissolve clots, and imaging that let us see the disease earlier, angioplasty and stents that mechanically reopen arteries. But also critically, we have device therapies. In the early 1980s, Michel Mirovsky, he implanted the first ICD, the implantable cardioverter defibrillator. And if you look at that photo, the device is absolutely enormous by today's standards. It's bulky, heavy, and crude, but it introduced the revolutionary idea that's a device inside the body that could detect and correct lethal arrhythmias in real time. So that moment marks a shift where cardiovascular disease went from being rapidly fatal to increasingly manageable. And it became a chronic condition that people can live with for decades. So how do they work? Let's zoom all the way in to where this surface engineering really matters. This is an ICD lead sitting inside of the heart. Its entire job is to sense the heart's electrical activity and intervene when the signal goes wrong. Those electrodes, the part that does the sensing is not the wire or the device. It's the electrode surface. So those electrodes are coated with titanium nitride, that brown-looking coating. Now, titanium nitride dramatically reduces the impedance of the electrode and increases its sensitivity to tiny electrical signals coming out from your heart. If you look at it under an electron microscope, you can see something pretty remarkable, and that's the surface is covered in tiny pyramids. In cross-section, you can see that actually the tops of micron scale columns. Those columns are porous. They're incredibly high surface area and fully accessible to physiological fluids. And that means the electrode doesn't just conduct electricity, it exchanges charge with the body in a capacitive way across a huge effective surface area. So with just a few microns of thin film, we can completely transform how that electrode performs. This is a perfect example of how thin films don't change the device, they change the biointerface. With more than 200,000 of these devices implanted globally every year, and a mortality risk reduction effect of around 45 to 55%, these devices have definitely saved lives. Lighteum Medical, we've made millions of titanium nitride coated electrodes for cardiac devices, and the impact this has had is a source of great pride for us. If you're interested in materials like this or something similar, then please get in touch. So these devices have had a major impact, but they're still evolving. They're becoming better. The next major evolution is extravascular ICDs. So this is where the lead is placed outside of the heart and outside of the veins. It's in a substernal position, and instead of running through the venous system into the heart, the electrodes are mounted externally. And this is a huge deal because almost all of the long-term problems with traditional transvenous ICDs, they come from putting a permanent wire inside of the heart and the bloodstream. Vascular damage, infection risk, mechanical lead failure, and extremely difficult extractions years later if you need them. The Achilles heel of ICDs has always been the lead. Now, they tried to solve this with subcutaneous ICDs below the skin. So this removed the vascular lead entirely from the heart. But it reduced the risk, but at a cost. Because the leads were located further away, there was no ability to pace, no pacing possibility, and there was very high shock energy required. So the battery life was shorter as a result. Now, extravascular ICDs, they thread the needle. They keep the lead outside of the vasculature, avoiding all those risks we talked about earlier, but they're close enough to the heart to enable pacing and sensing, similar to a traditional ICD. And this is where thin films is quietly becoming one of several key enabling technologies. These high charge injection coatings, like titanium nitride we just looked at, they make these electrodes so sensitive and efficient that they can reliably sense and stimulate the heart without being inside it. This wasn't possible before. A major milestone came in October 23 when the first EV ICD was approved by the FDA. It's a really great example of how advances in surface engineering, they don't just improve devices, they enable entirely new device architectures. Let's switch gears a bit and move from the heart to the nervous system, but keep the same idea, electrically enhancing coatings at the biointerface. Neural stimulation is simply the use of electrical signals to interact with nerves. either to stimulate them or to record from them. And this is happening all over the body. So deep brain stimulators for Parkinson's and tremor, spinal cord stimulators for chronic back pain, vagal nerve stimulators for epilepsy and depression, and sacral nerve for bladder control. And also cochlear implants that directly stimulate the auditory nerve to restore hearing. These 5 device categories dominate clinical neural stimulation today. And they all share the same requirement: extremely sensitive, stable electrodes that can both deliver and detect very small electrical signals in a biological environment. This is why the same types of high surface area, low impedance charge injection coatings we saw on ICD leads are so relevant here. Because in neural tissue, the signals are even smaller, and signal loss in the background noise is a really high risk. This market is growing really quickly. It's projected to grow from about 8 billion today to over 25 billion by 2034. So just look at where these electrodes are placed: in the brain, around peripheral nerves, along the vagus nerve. These devices are interacting with individual bundles of neurons in incredibly delicate tissue. What we're trying to record here is an extracellular action potential, the firing of a single neuron. And those signals are tiny, often around 100 microvolts. That's a very small signal sitting in a very noisy biological environment. To reliably record a single neuron firing, the electrode's geometric surface area needs to be extremely small, typically on the order of only a few thousand square microns, sometimes much less. If the electrode is too large, you stop seeing individual neurons and instead just record background neural noise. So we want very small electrodes. But here's the problem. When you make an electrode physically small, its electrical impedance shoots up. High impedance means more noise and worse signal quality, which is exactly the opposite of what we need. So this is where thin film coatings become essential. By coating the metal electrode with materials like titanium nitride, iridium oxide, or PDOT, we create a surface with enormous effective surface area at the microscopic level, while keeping the electrode geometrically tiny. That means we can keep the electrode small enough to listen to small groups of neurons, while giving it the electrical performance of a much larger electrode. So in other words, the coating allows us to decouple geometric size from electrical performance, which dramatically improves the signal to noise ratio for neural recording. This slide shows just how dramatic the effect of these coatings is. On the chart on the left, at the top, is charge injection capacity for different electrode materials. Bare platinum iridium coatings. Compare the bare platinum iridium to charge injection coatings like iridium oxide, titanium nitride, and especially PDOT. The difference is not incremental, it's orders of magnitude. Higher charge injection capacity and lower impedance. They mean 2 things. First of all, for stimulation, we can safely pass enough charge into the tissue without damaging it, even from very small electrodes. Secondly, for recording, lower impedance directly reduces electrical noise and improves high frequency response. This means clean recordings of those tiny action potentials. This is what allows us to build arrays like the one shown on the right. Hundreds or thousands of tiny electrodes, each small enough to talk to a very localized group of neurons independently. And this is a prerequisite for things like adaptive deep brain stimulation or functional stimulation after a spinal cord injury. and ultimately brain-machine interfaces like the Neuralink electrode array, which is shown on the right of the screen. So none of these systems are possible with bare metal electrodes. It'd simply be too noisy or too large and unable to inject enough charge safely. It's the thin film engineering at the surface that enables electrodes small enough, sensitive enough, and safe enough to interface with the nervous system at the level of individual neurons. So up to now, we've talked about coatings that help devices sense and stimulate. But there's another surface interaction that's just as important and often far more dangerous, and that is infection. Healthcare costs associated with healthcare associated infections are massive. They're a huge clinical and economic burden. In Europe alone, millions of patients are affected every year, with tens of thousands of deaths and billions in costs. Importantly for us, about half of hospital-acquired infections are associated with percutaneous access or implanted devices. That means the device surface is not just part of the problem, it's one of the biggest intervention points we have. The biological challenge is something called biofilm formation. Bacteria don't just land on a surface and they're vulnerable. They attach and begin to multiply, and then they build a protective matrix around themselves. Once the biofilm forms, the bacteria inside becomes dramatically harder to kill, dramatically harder than free-floating organisms. Antibiotics struggle to penetrate, and the immune system can't easily clear it. Clinically, this is why infections on grafts, pacemakers, prosthetic joints, and shunts often escalate to prolonged therapy, or worse, complete device removal. So again, we come back to the same theme, which is what happens at the surface defines the outcome. So how do we actually fight infection at the surface? Broadly, there are three strategies people use in antimicrobial coatings. First is like biocide releasing coatings, what you might call active release. These coatings release antibiotics, silver ions, antibodies, or nitric oxide into the local environment to kill bacteria. The second is contact killing surfaces. So materials that don't leach anything, but are chemically hostile to microbes on contact. Things like copper or certain quaternary chemistries, engineered oxide surfaces. The third is anti-adhesion or anti-biofilm surfaces. So coatings designed to stop bacteria from attaching in the first place. Polyelectrolyte multilayers are a great example, where you can engineer the surface to be unattractive to microbes while still being friendly to human tissue. The challenge is that none of these are perfect. Antibiotic coatings, for example, sound great in theory, but most device surfaces don't bind antibiotics well. If you do get them to bind, they often wash off quickly after implantation. So you only get a short-term protection. There's a bigger issue as well, which is antibiotic resistance. That's why there's been a growing interest in bactericidal materials like silver, copper, and titanium oxides. Materials that kill bacteria through physical or chemical mechanisms that are much harder for microbes to develop resistance to. This is where antimicrobial thin films move from theory into real commercial products. A great example is silver ion leaching coatings made by PVD processes. On the left is the Smith & Nephew ActiCoat dressing. It's a wound bandage where nanocrystalline silver is sputtered deposited directly onto the fabric. When the dressing is applied, small amounts of silver ions leach into the wound environment, preventing biofilm formation and dramatically reducing infection risk. On the right is the same idea applied to hard implants. So Zimmer Biomet's ZNN Bactiguard coating on intramedullary nails. Again, a thin silver coating, silver containing coating rather, deposited onto the implant surface, and that slowly releases silver ions once inside the body. It's the same thin film technology, two completely different medical applications. And the mechanism is elegant. Silver ions interfere with the bacterial metabolism and replication, making it extremely difficult for biofilms to establish on the surface. What's important here is that this isn't experimental. These are products in the market today, and you can see that the market growth for both silver wound dressings and antibacterial orthopedic implants is quite healthy. So thin film coatings can directly translate into reduced infection rates, better outcomes, and large commercial impact. Okay, so let's switch gears again. Let's change from biological surface function to a very different but equally critical requirement, which is visibility. During many minimally invasive procedures, clinicians rely entirely on fluoroscopy to see where a device is inside the body. The problem is that many devices, especially catheters and polymer components, they're almost invisible under X-ray because polymers simply don't attenuate radiation well. Historically, the workaround has been to add dense elements like platinum, gold, tungsten, tantalum, either as markers or blended into the material. On the right, you can see a nitinol stent coated with increasing thicknesses of platinum. The thicker the coating, the more visible it becomes under fluoroscopy. So the first instinct is simple, right? Let's just add a dense metal coating and the problem is solved. But as we'll see on the next slide, it's only half the story. So let's take a look at that platinum coated stent. When the stent is actually used, we see there are some problems. A stent isn't a static object. It's got to compress, flex, expand, and strain during deployment. Platinum is dense and radiopaque, and mechanically it's quite ductile, but it doesn't have anywhere close to the same elasticity as nitinol. So when the nitinol stent is strained, the platinum coating ends up cracking, delaminating, and spalling. Even more concerning, if you look on the chart on the right, mechanical testing showed that the presence of the thin platinum layer actually changed the radial force behavior of the stent. After compression, it didn't fully recover, and it required more force to close. So the coating solved the visibility problem, but broke the mechanical performance of the device. However, you may recall that PVD techniques give us a lot of control over the microstructure of the coating. It allows us to engineer films with a remarkable array of properties. So we ask ourselves here, how should this material be structured? Using advanced PVD sputtering, you can create a nanostructured columnar tantalum. If this coating on the left looks familiar, it should. It's very similar in concept to the columnar titanium nitride we saw earlier. These tantalum columns are disconnected from each other. They're not a solid brittle layer, but they can move independently. Think of blades of grass moving independently from each other. And what that means is this coating can stretch, it can flex, and accommodate strain as the stent expands and contracts, while still providing excellent radiopacity. So now we have a coating that is highly visible under fluoroscopy, does not crack or delaminate, and crucially, it doesn't alter the mechanical behavior of the stent. So this is a really nice example of how microstructure engineering, and not just material choice, can make thin films viable for a demanding medical device application. So now we're going to end with something that feels almost like it's like science fiction, but it's very real. And this is the ultra nano crystalline diamond, or UNCD. This is grown using microwave assisted chemical vapor deposition. And unlike conventional diamond coatings, the grain size is incredibly small, just like 3 to 5 nanometers. And that nanostructure creates a film that is ultra smooth, dense, and perfectly conformal, even on complex shapes. So from the materials perspective, this material has got so many advantages. It's extremely hard, it's wear resistant, it's got very low friction, and it's chemically inert. It doesn't corrode in body fluids, it's stable for long periods of time, and because it's pure carbon, it's also highly biocompatible with very low inflammatory response. Here's where it gets really interesting for bioelectronics. By slightly changing how it's grown, this material can be made to be insulating or semiconducting or electrically conductive. So this one material can be a mechanical protector, it can be a chemical barrier, or it can be an engineered electrical interface. One striking example comes from the work done by Professor Orlando Auciello and collaborators at the Argonne National Laboratory. They used UNCD to encapsulate silicon retinal implants. This ended up becoming the Argus Bionic Eye program. The UNCD coating reduced electrical leakage by over 2 orders of magnitude compared to bare silicon, while forming a pinhole-free biocompatible layer. They found that when they implanted this into a rabbit eye for months, there was no inflammatory response. In other work, modified UNCD surfaces have been shown to promote bone integration, supporting stem cell growth and appetite formation, meaning this same film can also help orthopedic implants integrate with the body. And that's a perfect example of how far thin films have evolved. All the way from the decorative gold leaf we talked about thousands of years ago to enabling the bionic eye. I think that's really incredible. So throughout this talk, the theme has been simple. The surface is where the device meets the body. Coatings let us tailor that surface in ways the bulk material simply can't, whether that's adding lubricity, electrical performance, antimicrobial behavior, radiopacity, biocompatibility, hardness, or even optical and dielectric properties. And importantly, we only need to modify microns at the surface to completely change how a device behaves. Looking forward, 3 forces will make coatings even more critical. First, miniaturization. Devices are getting smaller, less invasive, and more complex. Yet they still need the same durability, performance, and biocompatibility. Coatings are going to help designers to shrink devices without sacrificing function. Second, infection prevention and the economics of reducing hospital-acquired infections. They're just so enormously costly, medically and financially. Reimbursement pressure is driving demand for non-biofouling, antimicrobial, and drug-eluting surfaces. This is going to be one of the fastest growing areas in coatings. And thirdly, entirely new possibilities. Microelectronic implants, smart sensors, regenerative medicine, tissue engineering. These future devices will integrate sensors, actuators, and surface engineering into intelligent systems. None of that happens without advances in biomaterials and perhaps thin films as well. So coatings, they're not just an add-on, they're a foundational design tool for the next generation of medical devices. Thin films quite literally are enabling big impacts from the surface up. Thanks for listening.